Good morning, Professor.
Hello, when you speak, you always remind me I have to turn my volume up or I can't hear you. All right, that was Steve. Let me make a note of that. He's Steve. Not
SD, yes.
that's Steve. All right, well, let's start. We have only a 60-minute period today, and I have things that I absolutely have to cover. To remind you, if you haven't already done it, there's a quiz today, quiz 3, and it's available right now, will be until midnight. And another reminder, Monday is test 1. That will be a full period. You'll have all day Monday from midnight to midnight to complete the test. And therefore, there's no class on Monday. So don't show up for a Zoom session. Just sign in and do the test.
Professor?
Yes.
Hi, I have a question about the exam.
Yes.
So on exam, I just saw so that there will be any written question also.
The questions on the test will be the same as what you've been getting on quizzes. The same mixture. So there'll be some multiple choice, multiple answers, true and false, essay, matching. And it will cover everything, including there will be a couple of things from today. And so I want you to pay close attention today, and I'll try to point out things that, that you should pay special attention to.
Okay, thank you.
I have a quick question, Professor.
Yeah,
Does the quiz today just cover chemistry?
quiz today covers chemistry, biomolecules, and DNA, RNA, genes.
Okay, thank you.
So it covers everything up, including Monday of this week.
All right, thank you.
All right, I'm going to share my screen with you and go over the worksheet that you just completed. Hopefully you can all see that. Well, most of you did very well on this worksheet. And I'm just going to go over the answers quickly for those of you who might have missed a couple of things. Let me know if you can't see this. I'll make it a little bit bigger. There. DNA molecule is in the shape of a double helix. Monomers in proteins are amino acids. DNA is double-stranded. RNA is single-stranded. 4 classes of biomolecules: nucleic acids, proteins, carbohydrates, and lipids. What is a gene? It's a piece of DNA that encodes another molecule. Genes are on chromosomes. The 4 nitrogenous bases are adenine, thymine, guanine, and cytosine. Cystic fibrosis gene is CFTR, the transmembrane regulator. What's the meaning of this code? It indicates a gene location. Lipids that can act as hormones are steroids. Carbohydrates supply the cell with energy. This formula is for glucose. And a nucleotide has 3 components: a phosphate, a sugar, and a base. Males have X and Y chromosomes, females have 2 X's. Each human cell has 46 chromosomes. Some of you said 23. That's the haploid chromosome number. And the molecule in the bilayer is a phospholipid. So I said most of you got most of these right and scored well on this. Okay, if there's no questions about that, I'll take it down.
I do have one question.
Okay.
This is Anthony Emilo. For the top right, what are the 4 different classes of biomolecules? I put nucleotides, amino acids, monosaccharides, and fatty acids. Was that maybe for something else, or?
Well, that that would be the answer to something. I'm not sure what it would be on this grid.
Yeah.
But you see what the question was really calling for 4 different substances.
Got you. Okay.
Anything else?
I also have a question. This is Aiden Hensley.
Speak up, I can't hear you.
I also have a question. This is Aiden Hensley.
Ainsley, okay.
For the one formula for carbohydrate called, could galactose and fructose work as well? They share the same.
Yeah. And if you gave a different sugar besides glucose and I didn't give you credit for it, let me know.
Okay.
Professor, this is Alicia Carroll.
Okay, who's speaking?
I do have a question, Alicia Carroll.
Okay.
For on the bottom, it says human males have an X and Y chromosome while females have 2 X chromosomes. Do you usually write 2X or can you write XX, or does it not matter what?
Doesn't matter.
Right?
Either, they're both right.
Okay.
Professor,
Yeah.
this is Anthony Raban. Could you explain the code for gene location?
Yes. So, excuse me, this, we went over this the last time. I don't have the slide up right now, but this is a code for the location of some gene. So the 4 indicates the chromosome number. So this gene is on chromosome 4. The P indicates that it's on the short arm. Chromosomes each have a centromere that divides the chromosome into 2 parts, which are unequal in size. The long arm is called Q, and the short arm is called P. So this gene is on the short arm of chromosome 4. Then the numbers indicate particular bands, and the bands are due to specific stains that cytologists use to visualize chromosomes. So the 2 is band 2, which is quite large. 1 is subband 1 within band 2, and then 0.3 is an even smaller band where the gene is located. So that's why I say that the short answer here is this code is the gene location.
All right, thank you.
Professor, this is Steve Stenson. I just wrote out everything.
You wrote out everything?
Yeah, for the gene, everything that you just said, that's what I wrote.
Did I give you credit for it?
I don't know. I haven't checked yet.
Okay, well, if I should have given you credit for describing it. It doesn't have to be a brief cryptic note like I have here. So check, and if I didn't give you credit, send me a note and I will.
Okay.
Is there a question?
Professor,
Oh, sorry.
I have a question. My name is Priscilla. I was wondering, the slides of classify this set of slides to answer this is not showing up on my modules. Is that happening to anyone else? It says it might be corrupted or of an unknown file format.
Anyone else have a problem with that?
I couldn't find it on modules, but it's under files.
I didn't even see this under modules. I just went to assignments.
I usually go between modules and assignments, and I'll usually find it.
I don't find it there. I just find it in my calendar.
What is that?
Okay, Priscilla, does that answer your question? Usually, if you don't find something or you find it unavailable in one place, it could either be on the home page in a module, or it could be in a file.
I just looked it up under files and it says the same thing. It says this file cannot be previewed. It might be corrupted or of an unknown file, but it's fine. Okay, sorry.
I found some question on the book too.
You had a problem opening that worksheet?
Oh, me? No, the worksheet, I did the worksheet and everything. I just couldn't find the slides for it. So I had to go back and watch all of the videos and go one by one.
Oh, I see. And so is there just that one slide set that you're having a problem with, or all of them?
Yeah, it's just that one.
I think you can find it on the files. I did find it there.
Yeah, it says the same thing. That's why. Under files and the other one.
Okay. All right.
Thank you.
Yeah, I try to keep the files updated. Usually I download or transfer files in, and then in preparing for a class session, I update them or add things or delete things. Anyway, they're changed. And so I'll delete the original file that I deposited and I'll replace it with one. And sometimes that process does get corrupted. So let me know when it happens.
On here, just two quick questions. So CFTR stands for what again?
Cystic fibrosis transmembrane regulator.
Regulator, okay. And when we're looking at 4P21.3, could you break down what the location was? So like the 0.3 was on the short strand, right?
In this case, the gene that this code is specifying is on the short arm.
And what signifies that it's on the short arm?
P.
The P
stands for petite.
Okay. And if it was on the long one, what would it be?
Q.
Q. Okay. Thank you.
If you go back to the slide set from Monday, it's this is all laid out for you for the CFTR gene.
Honestly, I understood everything from that challenge grid, but that meaning of 4P21.3 confused me.
Yeah, well, that's what it is. And you can go back to either the slide set or the recording from Monday's session and get my full description of it.
Professor, I have a question.
Yeah.
Is it okay if we wrote 23 pairs of chromosomes in the nucleus instead of 46 chromosomes?
23 pairs is correct.
Okay, thank you.
Okay, anything else? All right, and I'm going to take this down.
I have a question.
Yeah.
My name is Bianca Olivas, and my question is related to the test on Monday. it will be like a time limit test or unlimited time?
It's a 75-minute time limit.
Oh, okay. Thank you.
So it's a full class period.
Quick question, Professor. I'm Alex Ellis. Is this slide going to be covered on the test one?
What I'm saying today, the slide set?
Yeah.
Yeah, it will be, there'll be a couple of things from this slide set on the test Monday, and I'll try to remember to point them out as we go along. All right, so we're going to talk today about DNA, RNA, transcription, translation, which is the equivalent to protein synthesis. And the case in point here is this CFTR protein. You see a diagram of it here. It's a complex protein that is transmembrane. That's where the T comes from. And it transfers chloride ions. And we'll go into a lot more detail about this later. But for right now, we're going to talk about the basic processes that have to happen in order for a protein like CFTR to be made. So first, we're going to talk about the replication process of DNA. Well, every time a cell divides, and you have millions of cells in your body dividing right now, and so they're copying their DNA furiously. But replication is copying only. Transcription of DNA to RNA, translation to protein are different and separate from replication. So separate these things in your mind. We're going to talk first about how DNA copies itself and then how it gets translated into protein. So DNA replication is divided into steps. And the first thing I want to do is show you this video, which outlines the steps quite clearly.
So now we'll go through a detailed step-by-step illustration of DNA replication. And what I've done here is I've listed all of the enzymes and proteins that are involved in this process. All of them serve an important purpose. And so we'll go through step-by-step in order to see how each of them comes into play. So the first step that happens is something called DNA helicase comes. And what it does, it's a 6-piece protein structure. You probably don't have to remember that. But essentially what it does is it unwinds the double helix. So here we have the 2 strands of DNA. And the helicase comes in and it essentially separates these strands. When it does that, these 2 strands serve as template strands. And the process is semi-conservative. That's an important vocabulary word. And what it means is that when we build new strands, we're going to build a new strand here, and we're going to build a new strand down here as well. The original DNA becomes the template strand for each of these pairs. And it's semi-conservative because each new double-stranded DNA segment contains half of the original DNA and half of the new DNA. So it's not fully conservative because you build new components, but it is semi-conservative because half of the new double-stranded DNA is the original DNA strand. So when helicase comes in, it separates the 2 strands, and this forms what is called a replication fork. The replication fork is obviously fork shaped like that. And what will happen is the replication will now be allowed to proceed on these strands because we have separated them and thus allowed other enzymes to have access. So helicase, it unwinds the helix essentially. It takes the helix and breaks it into its two substituent components. And those two components then serve as the template strand for the rest of the replication process. Next come single-stranded binding proteins, or SSBs. SSBs are necessary because remember that there is a strong favorable interaction between the base pairs on one strand and the base pairs on the other strand. Remember, these are complementary to each other. And so there is an incentive for them to get back together and bind with those hydrogen bonds. So in order to prevent that from happening, we have these small tetramers called single-stranded binding proteins, SSBs. And the SSBs essentially stabilize these so that they don't end up annealing, joining back together. And that's very important. They prevent the annealing so that these two stay separate. And some of them will show up down here like this. Essentially, they just bind to each template strand, each single stranded template component. And this is important because it prevents the annealing and thus allows these two strands to be separate and allows the replication process to continue to move forward. And so SSBs are there to stabilize the strands and prevent the annealing. As the process continues, as the primase and polymerase come along, they'll simply be displaced as soon as the polymerase shows up. But these are necessary in the earlier parts in order to make sure that these two strands don't come back together after they've been separated by the helicase. So we've gotten the first two out of the way. The helicase separates the strands, and then the SSBs come in and they maintain them in separate components so that they don't come back together and anneal or re-anneal. And then what happens is that DNA polymerase doesn't just start out of nowhere. What it needs is a primer. And a primer is a short segment of RNA. It's a small segment of RNA that will show up, and it will be small numbers. It could be 5, it could be 15 base pairs long. But essentially, a primer is a short RNA component that allows the polymerase to come and bind. So here we'll draw a primer here as well. So the primase shows up in order to produce these short RNA primer segments. And these primer segments then allow for the polymerase to come and start the replication process. So primase creates these RNA primers. They're required to start the replication. And then the big player, which is DNA polymerase, will come along. Remember that the directionality of replication is read up and write down. And so when it's reading from the template strand, it's going to be reading in the 3 to 5 direction. So this primer here will allow replication to continue in this direction. This one, because this is the 3 end and this is the 5 end on this strand, will allow replication to continue in that direction. So replication always starts at the 3 end and moves toward the 5 end of the template strand. The next step then is that DNA polymerase arrives. And this isn't an exactly faithful depiction of DNA polymerase, but you get the essential idea that it's something that connects to the template strand. And what it will do is it will essentially move along and produce more and more DNA. And so the polymerase will move in this direction and it will add bases that are complementary to our template strand. Every time that it encounters one of these SSBs, the SSB will simply be displaced and move away. So here's our polymerase on this end, and it will come along and it has a little attachment there. And essentially this will come along and it will continue to produce nucleotides that are complementary to the parent or template strand. And again, when it encounters the SSBs, they start to disappear. And this process will continue for a while. And so it will just continue to move. Remember that it reads in the 3 to 5 direction, but the new strand will be produced in the 5 to 3 direction. So this will be the 5' end of this, and the new strand will have a 3' end over here that it will continue building toward. Whereas here, the 5' end will be first, and it will continue to build toward the 3' end of the new strand. Remember that this new strand will be anti-parallel to the template strand. So what is the 3' direction of the new strand? It will be going in the 5' direction of the template strand. And so this will continue to happen, and it will build on more and more bases for quite some time. And then we'll get to a point where we encounter one difficulty. And so we'll just build this up to the end or so like that. What is happening here is that as this moves along, the helicase will continue to unwind the 2 strands. And as it does that, what you'll notice is that it will separate. So now let's just say that our parent strand is getting wider. Notice that this polymerase can continue to build into the strand. It will continue to produce new bases, and because the unwinding is simply going to be opening up further components, this top strand can just have the polymerase continue to move, and it will just be producing more bases. The issue is that this other strand, as the fork continues to open and it continues to separate these 2 components, notice that the polymerase is moving this way, and it will expose all of these base pairs that the polymerase isn't able to encounter. And so what will happen is we'll open it up more, and we'll need to build a new primer, and a new polymerase will need to come in. And so we'll get into this discussion of the leading and lagging strand, but recognize that there's a strand that is leading. And what that means is that one primer is necessary, and the polymerase can continue to build as the replication fork opens. The lagging strand is going to open, but notice the polymerase is building in the opposite direction. So these new ones, as it opens, will not get dealt with by this polymerase. And so this is called the lagging strand. And what it requires is it will require, again, another primase to lay down a primer and then to continue that process. And so next we'll get into a discussion of the leading and lagging strand. And that will then bring us into a discussion of RNase H, which is also known as DNA polymerase 1, and DNA ligase. And these are pretty much only necessary for this lagging strand that cannot continuously build as the replication fork opens. And so we'll discuss that. We'll draw this component. We'll redraw it so that the lagging strand becomes clear. And then the discussion will be complete, and you'll understand the replication process, how it's fairly continuous in this way, but it's discontinuous in that direction. So what we've done here is we've now advanced this replication fork. So the helicase has continued to unwind these strands. And so it opens up a new region of these 2 template strands that can now be replicated. And so on the leading strand, it's clear that because the polymerase is moving in this direction, remember it reads from 3 to 5, it can just continue to move in that direction into the replication fork and lay down more and more nucleotide bases. The issue emerges with the lagging strand. The lagging strand has a polymerase that's moving from the 3' end to the 5' end. But what's being opened up is further toward the 3' end than our primer is. And so what we need to do here is we will need to lay down another primer in the lagging strand. And then from that primer, we'll end up getting a new polymerase that shows up. And this polymerase will build on these bases up until it gets to the point where it meets the previous primer. This is called an Okazaki fragment. And it is something that only occurs with the lagging strand because the replication fork is opening and exposing bases from the template strand that are on the opposite side of the direction that the polymerase is going. And this is a discontinuous process, whereas the leading strand can continuously build more and more nucleotides that are complementary to the template strand as the fork opens. The lagging strand is unable to do that. And so you end up with a lot of Okazaki fragments. And this necessitates two other enzymes that we will encounter. And so the first one that we'll deal with is RNase H, or it could be called DNA polymerase 1. And the job of that is to simply remove the primer as the DNA polymerase 3 approaches this old primer. And so what it will do is the RNase H will then essentially eliminate that primer. And then the polymerase, the DNA polymerase 3, will continue to move forward up until it reaches the existing part of the DNA strand. But DNA polymerase 3 by itself is incapable of joining with an already existing strand of newly made DNA. And so what that requires is when the polymerase reaches here, you now need to find a way to link the sugar phosphate backbones of this already existing DNA with the newly synthesized DNA on this lagging strand. And so you need DNA ligase. Ligase is the same root word as ligature, and it essentially means to fasten or tie things together. And so you bring in a DNA ligase, and what that does is it simply, in an ATP-dependent manner, joins these two components together. And so now you're building a continuous strand based off of your template DNA strand. And this will continue to happen as the helicase keeps unwinding this DNA. What will happen is now we will need to lay down another primer, and then we'll build another Okazaki fragment, and it will continue to happen. So the lagging strand requires the use of RNase H and DNA ligase. And that is a consistent theme with the lagging strand. It's a very discontinuous process. The leading strand will only need RNase H. Remember, that's also DNA polymerase 1. It will only need it once because the polymerase 3 is just continuously laying down bases in the direction of the replication fork. So that's the big distinction between the leading strand and the lagging strand. And notice that the process of DNA replication is semi-conservative because we're building a new strand, but we're conserving the parent or template strand. And so it's semi-conservative. Half of this new double-stranded DNA comes from the original double-stranded DNA. There's also another word that we have to use, and this is semi-discontinuous. What that means is that the synthesis of the leading strand is continuous. It just keeps going. The polymerase can continue to move from the 3 to 5 direction. Remember that it's producing this new strand starting over here with the 5' end and going over to the 3' end here. It can continue to do this. And so it's continuous there. But the other half of it is discontinuous. It has to lay down new primer, produce an Okazaki fragment, get rid of that previous primer, and then join the newly synthesized strand with the previously synthesized one using a DNA ligase. And so that is a discontinuous process. So half of it is discontinuous and half of it is continuous. And so that means that the DNA replication is called semi-discontinuous because half of it is discontinuous and the other half is continuous. And as long as you understand that the lagging strand necessitates laying down more primer, having polymerase build more of these Okazaki fragments, and eventually joining them to the other bases we've produced with the DNA ligase, then you'll be able to understand the semi-discontinuous nature of DNA replication. And remember, this is what happens when cells are dividing in, for example, mitosis. And so DNA replication is something that you use not when you're trying to move from DNA to protein, but instead when you're trying to replicate DNA. And usually that is when you're forming new cells or having cells divide and thus more DNA will be necessary. But remember the leading and lagging strand, remember the different enzymes that can be involved, and remember the semi-discontinuous nature of it, that one of them is discontinuous and the other can just continue producing new bases all the way as the replication fork opens and exposes more of these template strands.
All right, who would have thought that copying could be so complicated? I'm going to share my screen again with you and we'll go back to the slides. All right, so we've just watched this video and this illustration shows some of the highlights from the video. A video is a lot and it takes a lot to digest, and so I'm going to try to break it down a bit more for you with the next few slides. First of all, notice that he put a lot of emphasis on 5' and 3' ends. And before we go further, let me tell you what that means. So that each strand of DNA has what's called a 5' end and a 3' end. Where do these numbers come from? Well, the carbons in ribose or deoxyribose are numbered from 1', 2', 3', 4', and 5'. Now, if the 5' and the 5' is always connected to a phosphate, but then it's usually then connected to the next ribose. But at the end, there's a free phosphate group. And that free phosphate group is a signal to enzymes that this is the 5' end. There's a free phosphate, it is the 5' end of the molecule. At the other end of the molecule, at the 3' position, if there's just another nucleotide, the enzymes don't notice that. But if this is a hydroxyl, they do. And they know then that this is the 3' end of the DNA molecule. And so you see that here on the left, there's a free phosphate at the on the 5' carbon, which indicates that's the 5' end. At the 3' end, there's a free hydroxyl, and that indicates that's the 3' end of the DNA molecule. And the other strand is just the opposite. It runs in the opposite direction. 5' starts at the bottom and ends at the top. So that's what 5' and 3' means. And why is that important? It's because enzymes have to know where they are. And in the case of DNA replication, they know that by recognizing, if they recognize a free phosphate, they know that they're at the 5' end of the molecule. If they recognize a free hydroxyl, they're at the 3' end. And this is important because DNA replication, as the man indicated, it always goes from 3' to 5 prime. And so he mentioned helicase, that's the unwinding enzyme, separates the 2 strands. Then the SSBs, which keep them from re-annealing. And then a primer is laid down. Here you see the RNA primer, and this indicates a point where DNA polymerase can come along and start laying down new nucleotides. And so as he emphasized over and over, the leading strand is continuous synthesis. You start with 1 primer on the leading strand, and the polymerase can go all the way to the end of the molecule. Has no problem. The problem arises because the new synthesis has to be 3 prime to 5 prime, and on the other strand, it's now the opposite. So as the unwinding occurs, the DNA polymerase can't proceed because it's on this strand, it's 5 prime to 3 prime. So it has to turn around and go in the opposite direction. So you have new primers laid down. The polymerase makes a short strand of DNA. This is called the Okazaki fragment. And then it makes another primer and makes another Okazaki fragment. And it just keeps going like that until finally it comes to the end. So, as I said, when you think of copying DNA, okay, you make a copy, but it's not like a photocopier. It has this very complicated process for making a new strand of DNA. And this happens every time a cell divides. I have this here just to emphasize 2 points. One is the difference between DNA and RNA. RNA has ribose, which means it has a hydroxyl on the 2 prime carbon. DNA just has a hydrogen on the 2 prime carbon. They both have a hydroxyl on the 3 prime carbon. And this is where the sugar phosphate backbone is joined, 1 nucleotide to the next, until the end, in which this carboxyl on the 3 prime is free. I've taken a screenshot of from that video to emphasize a couple of things. One is that these are the enzymes you need to know. And I've circled DNA polymerase 3 because this is really the essence of it all. This DNA polymerase makes new DNA from the old template, but it can't do it alone. It needs, first of all, to have the DNA unwound. Then it needs to have the strands stabilized. It needs to have an RNA primer on either strand before it can start. And then on the lagging strand, it needs an enzyme to remove the primer and another enzyme, DNA ligase, to join the Okazaki fragments. So when you study, keep these things in mind and try to keep in mind the names and the functions of each of these 6 enzymes. SSBs are not enzymes, they're just protein blockers. Now, I want to emphasize another point here on the lagging strand, and that is the Okazaki fragments. Okazaki is the name of the Japanese scientist that discovered them. So you have a leading strand and a lagging strand. As I said, DNA polymerase always goes from 3 prime to 5 prime. And so on a leading strand, it can start at the 3 prime end and it can go all the way to the 5 prime end, making new DNA as it goes, and it has no problem. The problem is on the lagging strand, it goes some distance, and then it's now going the wrong direction. It's going from 5' to 3'. So it has to turn around and go the other way. And it does that by laying down a new RNA primer, making a short segment of new DNA, again, going backward. And then it comes to another primer, and this is where the RNA H bumps this primer off, and then DNA ligase joins the two. So you see that here, this nick, the backbone is not joined, but DNA ligase, see it here, joins it. So Okazaki fragments occur only on the lagging strand. The DNA polymerase 3 builds from a primer on either strand. RNase H removes the primer this occurs only on the lagging strand. And DNA ligase joins the Okazaki fragments. And this occurs only on the lagging strand.
Professor,
Yeah.
it said Nick on the last slide.
Uh-huh.
Is that an abbreviation or acronym for something?
Well, it's a nick, like if you scratch yourself, you've nicked your skin.
Oh, okay. Okay.
And it's to me, it's not really a nick, it's a gap.
Right. That's what it looks like to me too.
Right, this is a close-up of what I've just been saying. You have the leading and lagging strands, and the Okazaki fragments are formed and joined by DNA ligase. Now, he made a big deal about the fact that DNA replication is semi-conservative. And this means that in the new strands, one strand is from the original old strand, that's the dark red. The pink is the newly synthesized. And so when DNA is copied, half of each strand is new and half is old. The contrast, if it were a conservative replication, then you'd end up with two molecules, one of which would have both strands old and the other both strands new. Well, this does not happen. What always happens is semi-conservative. Dispersive, we won't talk about because it's just hypothetical, never happens. So that is the end of DNA replication. DNA replication has to occur every time a cell divides. Cells are dividing all the time, and so this very complicated process is going on in every living cell most of the time. And now we're going to turn to how does information get from the DNA molecule to the protein. This involves transcription and translation. What is the protein? I'm going to go through this fairly quickly because this is all review. You remember the groups that all amino acids have, amino and carboxyl, and they're only distinguished by their so-called R groups. One thing I want to emphasize here, I told you before that two of the amino acids found in protein have sulfur in them. We'll come to that later. But there are other amino acids that have these hydrophobic groups, in this case, methyl groups. And these are important in forming bonds. They're not really bonds, they're really attractions that hold things together, even though they're not bonds. And I'll come back and talk about how these hydrophobic groups do that in a little bit. Peptide bond, we talked a lot about that. How a peptide bond has to be in this context, D double bond O and H. If it's any other context, it's not a peptide bond. And peptide bonds always occur in polypeptides and proteins, never occur in any other molecule. All right, information flow in cells. Now, this is an overview, and it has all the players here: DNA, RNA, and protein. So the big blue ball is a nucleus. In the nucleus is the DNA. When DNA is transcribed into RNA, again, the double helix has to unwind, enzyme has to go in and make a new strand, which is now RNA. The DNA cannot escape from the nucleus. It always stays there. It's never anywhere else. RNA is produced in the nucleus, but it leaves. It goes through these pores in the nuclear membrane and ends up in the cytoplasm, where it encounters other forms of RNA, ribosomal RNA and transfer RNA, and the three of them together make a protein. So that's the overview. Types of RNA, ribosomal. There are two types, a large subunit and a small subunit. Transfer RNA, there are 20 of those, one for each amino acid. You see that here. Messenger RNAs, there are thousands. Each protein in a cell has a unique messenger RNA. So what happens is that the tRNA has the three-letter code for a particular amino acid on one end of the molecule. The other end has that amino acid. So this three-letter, so it's called an anticodon, matches up with three-letter codons on the messenger RNA, so that one amino acid can be added after another. I want to emphasize here that as this transcription is going on, you see, whenever there's a T, the RNA gets an A, but whenever there's an A, if it were DNA, it would specify a T, but RNA does not have T's. It has U's instead. So I've circled this a U because that's what's different in transcription to RNA. So there are two types of ribosomes, prokaryotes and eukaryotes. But what happens here at the top left of this figure are the three types of RNA: mRNA, rRNA, and tRNA. So this is the message. mRNA is the message. Here you see a start codon, AUG, and a stop codon, UAG, the large and small ribosomal subunits, and a tRNA carrying a unique codon and an amino acid. The first step is that the ribosomal subunits clamp onto the message, and they focus. There's a signal that tells them that this is an AUG. This is where the protein will start. And so the tRNA that will be brought here carries a methionine. AUG is the codon for methionine. So the first amino acid in virtually all proteins is methionine. And then it moves to the next codon and it calls in another tRNA with a different amino acid. That gets added to the chain. The chain builds up. And finally, the ribosome comes to a UAG, which means stop. That's where it ends. So now we have a completed polypeptide, and the other components can go back, get recycled, and do it all over again. This you have to pay close attention to. You'll have to know this for the test on Monday. How can you determine the amino acid sequence? So we start with a particular sequence of DNA shown here. Now to copy that, if it just the cell dividing, to copy this, the T would specify an A in the new strand, an A, a T, a C, a G, and so on. However, to transcribe that now, not to DNA, but to RNA, the T specifies an A. The A would be a T, but now it's a U. C is G, A is U, and so on. And so now you have a message of RNA where there are no T's, wherever there would be a T, there is now a U. And now you want to know what is the peptide that this mRNA is specifying. That's called translation. And so you can do this, and the way you will do it is with a codon usage table. So you have a codon, AUG, and you say, what's the first base? Well, it's A. What's the second base? It's U. So that's this box. The third base is G. AUG is methionine. So the first amino acid in this protein and in most proteins is methionine. Then you go to the next one, which is UCA. So the first base is U, second base is C, third base is A, that's serine. So you write down second base, the second amino acid is serine. Third is AAU. So the first base is A, second base is A, third base is U, that's asparagine. And so you write down the third amino acid is asparagine. And finally, you come down to the last one, AGA. First base is A, second base is G, third base is A, AGA is arginine. You write that down. And finally, you come to UGA. First base is U, second base is G, third base is A, UGA is stop. That's, there's no amino acid here. Arginine would be the last amino acid in this peptide, polypeptide. So is this clear? And it looks easy when you watch me do it, but you're going to have to do this yourself.
Professor, when you were translating it, did you skip one?
Yeah, I skipped over. Yeah.
Why did we skip UGU?
For the sake of time. I'm running out of time.
Okay. But we would just go through the whole thing.
You go through the whole thing, yes.
And then for transcribing, it's really just take any T and change it to a U?
Well, you have to transfer, you have to do the complementary strand. So the DNA strand starts with T, remember that. And T, when you transcribe, will be an A in the transcript. And then the next letter is A, and that would be a T, except now it's RNA, and RNA doesn't have T's. So instead of T, you put in a U. And then the next nucleotide is a C, that's G. Next is A, that's U. Next is G, that's C. So it's complementary.
So, excuse me, we're gonna write that under the first one, the first row, like let's say.
Say it again.
Like we're gonna write them, let's say let T, we're gonna write under it, the A.
Yeah, so if I give you a sequence of DNA and I say transcribe that, you would do this.
Okay.
You would
Under it.
Put yeah, right under it. Put the complementary strand, making sure that it's RNA and not DNA.
When you need it, I'm gonna specify when you need it for the RNA, not for DNA.
Right. Right.
Okay. Professor, and do we put like, for example, AUG, do we write down met underneath?
Yeah, if the codon is AUG, under that, under the translate part, you'd write met, 3-letter code.
Okay, thank you, Professor.
And you'll have a codon table in the test that you can refer to.
So
It'll
we're
look
not
just like
doing this on the quiz today, right? It's for the test on Monday.
This will be on the test Monday, yes.
But there's nothing about like DNA and RNA on the quiz today, right?
No, that's correct.
Okay.
None of this in today's lecture is on the quiz today.
Okay.
A few things
And
like this will be on the test for Monday, but you have time to study for it between now and then.
I'm assuming we don't have a quiz next Wednesday.
No, there's no quiz next Wednesday. We'll move on to new material next Wednesday. Okay, and now we're going to talk about protein structure. There's primary, secondary, tertiary, and quaternary structures. Proteins have no activity until they're not only the right length, but folded properly. And to fold, several bonds are employed, and we're going to go over that now. So the primary structure of a protein is the amino acid chain. So this is just a chain starting at one end, amino acids to the other end, the carboxyl end. And this molecule has no activity. It's just a chain of amino acids. Now, that primary chain can coil into what's called an alpha helix, or it can fold into what are called pleated sheets. And both of these, the alpha helix and the pleated sheet, are held together by hydrogen bonds. So this is secondary structure. Tertiary structure folds in an even more complicated way. And I want to point out the kind of bonds, and these are intramolecular bonds. In other words, it's bonding from one part of the chain to another part of the same chain. And this is important, that a single chain bonds to itself back and forth, and causes folding, so that you end up with a very complicated structure like this. And I'll come back to that in a minute. So hydrogen bonds are critical. And you know what a hydrogen bond is between a hydrogen and an electronegative atom, like oxygen. These disulfide bridges, when there are two sulfur-containing amino acids in the right locations, a covalent, and remember this, the SS bond is covalent, so it's very strong, will form, and that will hold the chain in a particular configuration. And then there are ionic bonds, where positive and negative charges attract each other. And then there are the hydrophobic interactions. Now, these are not bonds, but they play a role in helping the protein to fold. And I'll explain what this means in a bit. In fact, I'll explain it right now. So you have these methyl groups, and each of these methyl groups is somewhat hydrophobic. That is, they don't like to be in water. And so consider these orange balls to be methyl groups. And each methyl group has 8 water molecules attached to it. Doesn't like that. The other one is the same. And so what they do is they're attracted to each other because they're both hydrophobic and they're trying to get away from water. And so whereas individually, they together have 16 water molecules, if they get together, they only have 10. And so they're much happier getting together and getting away from some of those water molecules. And that's what this attraction is. It's not a bond, but it's a force that helps to hold the protein, the polypeptide chain in place. And now the final type of structure is called quaternary. So a single chain can have activity if it's folded properly. But in some cases, one chain isn't enough. And an example of that would be hemoglobin, which we have in our blood, where it requires 4 different chains held together by hydrogen bonds and disulfide bridges, but the 4 separate chains held together in a complex has activity. So any one of these 4 or 2 of these 4 is not active, but you put all 4 together, it becomes hemoglobin, carries oxygen. And a quaternary structure can be either globular or linear. For example, collagen is an example of that. And this is the cover of Science Magazine last month. And the point here is, first I wanted to show you what a pretty folded structure this is. This would be an active protein. The point of this article was that there are now algorithms that will, within a few minutes, take an amino acid sequence, which you see here in these ribbons, are the amino acid sequence. And within minutes, it can predict the folding. Where you see the alpha helices, the beta sheets, and other forms of structure that are necessary in order for this protein to have activity. And so, primary structure summary. The primary sequence is the linear amino acid sequence. These are held together with covalent peptide bonds. Peptide bonds are found only in proteins. Secondary structure is alpha helix, beta pleated sheets. These are held together with hydrogen bonds. Tertiary structure are the only ones that have, or I should say the first, tertiary and quaternary have activity. Tertiary structure, a single protein amino acid chain folds and links to itself. And the bonds that help it do that are hydrogen bonds, hydrophobic attractions, and disulfide bridges, which are covalent and form between methionine and cysteine residues. And then there are the ionic bonds that can form. Quaternary always requires more than one chain. Quaternary structures have 2 or more chains that are linked together with hydrogen bonds and disulfide bridges. Okay, types of proteins. I'm not going to go through a lot of this today because I'm out of time. In fact, I'm over time. I just want to make one final point that I thought you might be interested in, and then I'll come back and cover this at the beginning of next lecture. But one type of proteins are structural. So for example, collagen and bones and skin and so on. Keratin, spider silk, which is very strong material. Then there are contractile proteins like actin and myosin, which make up muscles. And this raises the question, how do muscles actually work? So here's a stylized drawing of a muscle fiber, set of fibers. But how does it work? When you flex your muscle, what actually happens? Well, it turns out that your muscle fibers are made of these two proteins, myosin and actin. So the myosin fibers are these black bars. Actin is the narrow black bars. And if we start at number 1 here, you can see that this head on the myosin is tilted at about a 45-degree angle. Then in step 2, it is straightened so that it's now vertical. In step 3, the myosin fiber attaches to the actin fiber. And in step 4, the attachment again switches to this 45-degree angle. And in doing so, the myosin fiber remains stationary, the actin fiber moves from right to left. So it's like walking. When you flex your muscle, it's like the actin fiber is walking along with respect to the myosin fiber. And with that, I'm going to quit because I'm genuinely out of time, and I'll come back to this, start again here next time. So in the meantime, if you haven't taken the quiz yet, please do before midnight. And on Monday, the test 1 will be available to you, and we will not have class on Monday. Everybody okay with all of that? I know I've given you a lot today, so I hope you can digest it, and I've tried to tell you the things that you will need to know for the test on Monday.
So we'll see you next Wednesday.
See me next Wednesday.
Thank you.
Okay, bye. Have a good day.
All right.
Thank you. Hi, everyone.
Thank you.
Bye,
Thank you,
everybody.
Professor.
Have a
Good
good
luck on the
weekend.
test. Good luck on the test.
Yes, please have good luck. More than luck, because you know it.
Bye.
Bye.